We can get started. Hi, everybody. Welcome to the MGH Brain PBM rounds. Today is January 20th, 23, and it's my great pleasure to present to you Dr. Sadra Shahdadian from the University of Texas, Arlington, and from Cook Children's Hospital at Fort Worth in Texas. Sadra, thank you for coming in, and please take it away. I know you're going to talk about different target engagements today with photobiomodulation.
Hello, everyone. Thank you very much for the invitation. I'm very glad to be with you today. So let me start sharing my screen. Okay. Can you see my screen?
Yes, we can.
So today, I'm going to talk about the effects of transcranial photobiomodulation on neurophysiological network of human prefrontal cortex. I'm Sadra Shahdadian. These results are mainly from my PhD dissertation, which I was under the supervision of Dr. Han Liu and her associate, Dr. Jinlong Wang. So, of course, all of you know the mechanism behind the tPBM and how the light and tissue interact with each other. But just for a quick review, we have different types of light-tissue interactions. So, for example, the most common or the oldest way is the FNIRS. So we can use the functional near-infrared spectroscopy or FNIRS to monitor the hemodynamic states of the superficial cerebral tissue. The other way is the deep brain stimulation or DBS with the near-infrared photobiomodulation probe. And the one that we are talking about today is PBM, or in this case, tPBM, which is transcranial photobiomodulation. In our lab, what we do is mainly combining the methods 1 and 3, where we have tPBM light combined with FNIRS and different other modalities like EEG or other modalities. And the mechanism behind this effect of tPBM on the tissue is mainly, as you can see in the figure, this is borrowed from a paper, is that the light can be absorbed in the sensitive CCO or cytochrome C oxidase, and it leads to the production of ATP and nitric oxide. So the mechanism behind it is assumed to be similar to what we have for regular neuron activation. Where in neuron activation, we have the increase in glucose consumption, increase in oxygen consumption, and increase in ATP consumption, which leads to the increase in the cerebral blood flow and blood oxygenation. Or on the other hand, when we have the tPBM, which is called also as TILS, and it reaches the CCO and the mitochondria, it leads to an increase in ATP synthesis, which again leads to increase in the cerebral blood flow and blood oxygenation. Other studies have shown that we can see an increase in the oxygenated hemoglobin and cytochrome C oxidase, the redox state, during the stimulation and even after stimulation. Also, some other studies have shown how it can affect the electrophysiological networks in the brain. And for example, in this study, you can see that in the tPBM group, we have more connectivity between the prefrontal cortex, which is the stimulation side, to the temporal and parietal cortex. So the main brain physiological subsystems, we can focus on 3 of them, which are the electrophysiological, hemodynamic, and metabolic subsystems. And each of them are measured by specific methods. So for example, electrophysiological system can be measured or monitored through EEG rhythms. Hemodynamic is measured through the changes in the concentration of oxygenated hemoglobin, and also the metabolic can be measured by the changes in the concentration of redox state of the cytochrome C oxidase. So the biomedical signal from each subsystem can be represented as a time series, and the connectivity assessment helps to construct the topographical or physiological network. So topographical network or intrasystem network is the functional connectivity showing the information flow between different regions of brain within a specific system. And each region can be considered as a node, and the connectivity between each pair of nodes can be quantified and represented by weighted links. On the other hand, we have physiological network or intersystem network, which is the functional connectivity showing the information flow between different systems within a specific region of brain. So for example, in this figure, you can see that we have EEG, delta CCO, and delta HPO as different systems in the same region. And we want to see how these are interacting with each other. So each system can be considered as a node, and the connectivity between each pair of nodes can be quantified and represented by weighted links. So we have three main links here. The most popular one is the neurovascular coupling, which is the coupling between EEG and the changes in the concentration of HPO. The other one is the neurometabolic connectivity or coupling, which is the coupling between EEG and changes in the cytochrome C oxidase concentration. And the last one is the coupling between CCO and HPO concentration changes, which we call metabolic vascular coupling. So in the first set of data that we collected, we had a 1064 nanometer laser with 4.2 centimeter diameter, and laser power was set to 3.5 watts, and the stimulation site was right prefrontal cortex. We collected 45 subjects. We had the EEG data, 64 channel EEG data from them. The protocol included the sham-controlled study for all subjects. And then in each measurement, we had 2 minutes of pre-stimulation, 8 minutes of active tPBM or sham, and 3 minutes of post-stimulation. In the analysis, I split this 8 minutes of active tPBM or sham in two time segments, the first 4 minutes and second 4 minutes, which we call STIM 1 and STIM 2, to better see the dose-dependent changes in the modulation. So for the results that we get, the first one is the power modulation in the electrophysiological system. So as you can see, the main effect of the tPBM on the right prefrontal cortex is associated to the alpha and beta bands, which we can see an increase in the power in the frontal and parietal regions in alpha band during STIM 2 and post stimulation. Also, we can see an increase in the power of the beta band in the STIM 2 in the central and temporal regions. We also can see a decrease in the power of electrophysiological activities in the delta band, but other studies have shown that this decrease in the delta band, decrease in the power in the delta band, can be associated with the feeling of that heat on the scalp. So when we're talking about the connectivity, one of the ways to look at the connectivity is looking at the graph theory metrics. So we have nodal and global metrics. Today I'm going to just report the nodal metrics. So for example, when we're looking at a network and we look at one node in the network, we can measure the global efficiency as one of the metrics. The global efficiency shows the integrity of that node in the information flow in that network. On the other hand, we have clustering coefficient, which is the metric for the segregation or the clustering of the neighboring nodes and how they are segregated from the other nodes in the network in the information flow. Also, we have the betweenness centrality, which identifies the hubs of the information flow in the network. So when betweenness centrality for one node is higher than the other nodes, it means that this node plays a role of a hub in the information flow. And also we have the degree centrality, which shows how many connections do we have to this node from all other nodes in the network. So when we look at these and see how they change after tPBM, and we compare it with the sham stimulation, we can see that, for example, here we have 5 columns. Each column shows the nodal metric, and the rows show different stimulation temporal segments. So we have stim 1, stim 2, and post. So the first 2 columns show the integrity. When the color is blue in a region, it means that the value is decreased in tPBM compared to sham. So we can say that in the left frontal and right frontal regions, we have a decrease in the clustering coefficient and nodal local efficiency, which are the measures of segregation in tPBM compared to sham. So it means that in these regions, these nodes are less segregated compared to before stimulation. On the other hand, we have nodal efficiency and betweenness centrality, which are the measurements of the integration. It means when a region is red in this figure, it means that after tPBM, we have more integrated role of this region in the brain network, especially because we are talking about the resting state network. We can see that right frontal and left frontal regions are playing a very important role after tPBM, especially as a hub, which is measured by the betweenness centrality. And also we have the degree centrality, which is showing the number of links or significant links from the other regions to these 2 regions, right frontal and left frontal regions, are increased after the tPBM stimulation. The next study that we did was mainly using the BB-NIR system, which is a broadband near-infrared spectroscopy, which helps us to measure the changes in oxygenated hemoglobin and cytochrome C oxidase concentration. And
Sadra, can I stop you right there? I know this is kind of a new chapter here. I was intrigued by the differences in the prior slides in between the first and the second treatment. So it seems like the first treatment has effects that are more unilateral to the site of stimulation in the prefrontal cortex, and then somewhat it expands on both sides.
Exactly.
So I was wondering about that. And also, so this is related to the connectivity. And also when you looked at your alpha signal, it was much more pronounced after the second treatment, although you could tell right from the first treatment that there was an effect. So I think this data, I'm curious about your thoughts, kind of a very intuitive interpretation is that this data from a neurophysiological perspective, they kind of validate the clinical understanding that multiple sessions build on each other, at least clinically and seemingly neurophysiologically.
Exactly. So it shows the dose-dependent effects of tPBM, that we need to make sure that we have the enough dose and we have the enough repetition to make sure that it has the effect. So yeah, this study is mainly looking at the during stimulation and post stimulation, and it shows that even in post stimulation we still have the effects. So it's very promising, as you mentioned. Yes, so it starts from the stimulation side and then expands to the lateral side, the contralateral side.
Thank you.
So the next study was mainly focusing on the basal motions in the vessels in the prefrontal cortex. As I mentioned, we could measure it by BB-NIRs, and by having a time series, we could have different frequency bands, and as I will explain later, how we could separate these frequency bands from each other, and how to assign each of them to one layer of the vessel wall. So each vessel wall inside the brain has three main components. The first one is the endothelial layer, which is made of the endothelial cells. We also have the smooth muscle cells, and we have another layer that is responsible for the interactions between nerve endings and the vessel wall, and also it contains the fibroblast cells and other cells. So when we collect the data, especially Beginner's data, we can reach the very slow oscillation. So for example, if we have a full frequency data, in this case, we have a 7-minute time series of changes in the oxygenated hemoglobin. We can assign this to different frequency bands. We can transfer it to frequency domain and see the amplitude in different frequency bands or in different frequencies. So studies have shown that these frequencies or bands are corresponding to a specific layer of the vessel wall. For example, in the endogenic band, we have the frequency band of 0.005 to 0.02 hertz. And if we filter this part of the signal and regenerate the signal, we will see a very slow oscillation in the changes in the oxygenated hemoglobin. The other one, the next one, is the neurogenic band, which is responsible for the interaction between the vessel wall, the vessel dilation, and neurotransmitters and neuron endings. And the frequency or the oscillation in this band is a bit faster. The last one, which is the fastest in this range, is the myogenic band, which is corresponding to the 0.04 to 0.2 hertz, and it shows the oscillations in the smooth muscle cells. So by separating these three components in the time series, we can look at them separately and see which layer of the vessel wall is oscillating differently from the from before the stimulation. So the setup we had was a 2-channel Beginner's, including 2 main channels on the right prefrontal cortex and left prefrontal cortex. Each channel includes a light source and a detector. So the protocol included a 20-minute protocol. We had 7 minutes of Beginner's recording, followed by a 8 minutes of tPBM or sham on different locations of prefrontal cortex. And then we had the 7 minutes of recovery. The stimulation was randomized. We had 5 visits for each subject. In this study, we had 31 subjects. And the visits include the right 800 nanometer tPBM, left 800 nanometer tPBM, right 850, right sham, and left sham. So we had 5 measurements for each subject. So for the connectivity analysis, as I mentioned, we have 2 channels, and in each channel, we can detect 2 different time series, delta HBO and delta CCO. Sorry, I think we have something. Simultaneously, we're measuring the left channel, which is corresponding to the left prefrontal cortex. And again, we have changes in the delta HBO and delta CCO concentration. And then we can make a network of these 4. So for example, we have HBO on the right, HBO on the left, CCO on the right, and CCO on the left. So how we name them is that the connectivity between HBO right and HBO left, we call it bilateral connectivity of HBO. Similarly, we have the bilateral connectivity of CCO. And for the coupling between HBO and CCO, which is the coupling between hemodynamics and metabolism, we call it unilateral coupling of HBO-CCO, either on the left or on the right. So first of all, we measured it because we had a relatively good data set. We had 31 subjects and 5 visits. And before the stimulation, we had only rest. So we had a data set of 150 measurements for the rest. So we thought we might find relatively reproducible values for the connectivities especially bilateral connectivity and the unilateral coupling. So here in this figure, you can see the results for all the 135 measurements. We have the different frequency bands, namely endogenic, neurogenic, and myogenic band. And then we have the blue bar, which is the changes in the delta HPO. It's the bilateral connectivity of HPO. And the orange one is the bilateral connectivity of CCO. As you can see here, the bilateral connectivity of HPO is significantly higher than CCO because, as we expect, the hemodynamics is more global, but the metabolism or inside the mitochondria are very more local. It's not global. So that's why the oscillations in different regions of the brain are not well synchronized in the metabolism and concentration of CCO. Also, we have the unilateral coupling of HPO and CCO. So this coupling between HPO and CCO, we don't see many significant changes between left versus right. However, in the neurogenic band, we have a slight increase, or not increase, higher values for the coupling between HPO and CCO, which is actually HPO-CCO coupling shows the demand and supply coupling, or how it is balanced between demand and supply for the oxygen. So we see that in the left, we have a relatively higher values of this coupling on the left side. So we get to the tPBM induced modulation in bilateral connectivity. As you can see, because we had 3 different stimulations, we have different effects of each stimulation. So again, we have 3 different frequency bands and 3 different stimulations. So R800 means that we have right stimulation with 800 nanometer laser, L800 is left stimulation with 800 nanometer laser, and R850 means that we have right stimulation with 850 nanometer laser. As you can see, we see the differences between both wavelengths of the laser and the location of the stimulation. So as you can see here, for example, the right 800 nanometer laser increases the bilateral connectivity of HPO. However, actually in the endogenic band, however, the left 100, 800, left side 800 and right side 850 doesn't show this any changes actually. But in the myogenic band, where we're dealing with the smooth muscle cells, we see a decrease or decoupling or decrease in the bilateral connectivity between left and right. It means that the effect of this stimulation is local. When we have a decrease in the bilateral connectivity, it means that this effect on that specific region is more local than global. So we can see again the bilateral connectivity of CCO. In this slide, you can see that we have again an increase in the bilateral connectivity of CCO, which shows the metabolic synchronization on bilateral side. Right side 800 endogenic band can increase the bilateral connectivity, but in the neurogenic band, we have a decrease in the bilateral connectivity of CCO in the neurogenic band. Again, it shows that the effect of these stimulations are very local in this frequency bands, and in the myogenic band, we have different effects of different stimulation sites for right versus left. And other studies have shown that these right stimulation versus left stimulation can be very different from each other, but even by the behavioral tasks. So it needs to be investigated further how the different location of stimulation can affect the synchronization or the oscillation in each like for example, hemodynamics or metabolism. And we have the unilateral coupling.
Can I kind of stop 1 second here, trying to make sense of this data, and I definitely want to have your input, and maybe I speak for the crowd here. So I think one aspect that is interesting that might come out of this is that the brain has hemodynamic changes that can occur, and they can be fairly diffuse after even kind of having stimulated a specific area. However, the metabolic changes that are actually possibly related to the actual photodeposition and the photochemical effects seem to be somewhat accentuated in the area that is actually exposed to the light. So there might be a benefit kind of fairly global when you're treating even a single spot, but then a more specific benefit at that level. Is that kind of how you have interpreted this? Or I'm curious about, you're showing a lot of data here.
Yeah, exactly. So that what we're seeing, first of all, for example, endogenic, because it's talking about the endothelial layer. Endothelial layer is a part of the blood-brain barrier. So it's just the first step to look at this data and see how we can affect different parts. So for example, endogenic band, if we see an increase from right stimulation with 800 nanometer, we see a bilateral connectivity increase in the CCO. It means that the effect is global in the in specific frequency bands, and then it can be investigated further to see if, for example, if we shine the light on the right side, does it affect the blood-brain barrier permeability in one side or on the other side as well? On the other hand, we have neurogenic band, which is the interaction between the neurons and the vessel wall. It shows that because the neurons are mainly activated on the specific side of the stimulation, are affecting mainly that specific region and the vessel wall on that specific region. So we are mainly focusing on the similarities of different stimulations, which is, for example, the decrease in the bilateral connectivity. So the decrease in bilateral connectivity, as you mentioned, it is showing the effect. It is showing that the effect is more local than global. So what I'm trying to show is that we have different options from different stimulation. If you want to locate your stimulation on one side and just get the specific result from that side, you can choose between these different stimulations. But if you want to get effect globally or contralaterally, you can choose another type of stimulation. But this is just the first step. It needs to be investigated much further.
Thank you.
Can I ask a follow-on question?
Sure.
Why did you choose stimulation to the right frontal cortex as opposed to looking at other areas? Is there more of a intranodal connectivity through the right frontal cortex than other areas of the brain?
So first of all, we had these previous studies that we had in our group with 1064 nanometer laser, and we got some behavioral effects of right prefrontal cortex stimulation with tPBM that showed the increase in the working memory task and the improvement in the reaction time. So we wanted to see, and on the other hand, we didn't have any behavioral results from that specific tasks from the left stimulation. So we wanted to compare what the biological or physiological effect of these different stimulations are to help us to interpret the results of the behavioral tasks. So the last slide of this section is the unilateral coupling in the ipsilateral to the stimulation side. So the unilateral coupling shows the interaction between supply and demand of oxygen here, because it's metabolism and oxygenated hemoglobin. So in the endogenic band, as I mentioned, which is corresponding mainly to the blood-brain barrier or endothelial layer, we see an increase in the coupling between metabolism and the hemodynamics on the ipsilateral side to the stimulation. But we get different results from different stimulations on the other frequency bands. On the other hand, we have the unilateral coupling on the contralateral side to the stimulation that we don't see any significant changes in this coupling between hemodynamics and metabolism. So again, this slide and these results show that the effect of all three stimulations are mainly local. It means that the physiological effects on the hemodynamics and metabolism is not that significant on the other side. So the other thing that was interesting for us was looking at the EEG and Beviniers measurement, which shows us which adds another node to our measurement for our network, which is the electrophysiological activity. So the setup we had was similar to what we had for the other measurement, but it includes also EEG electrodes. Again, we had 22 minutes of experiment, including 7 minutes of Beviniers and EEG recording, followed by a tPBM or sham stimulation on the left side. And then we had, again, the 7 minutes of recovery time for the Beviniers and EEG recording. So here, as you can see, we have the physiological network. We call it physiological network because it's showing the interaction between EEG, CCO, and HBO in the specific site. And again, we have the different frequency bands, and we have left prefrontal cortex and right prefrontal cortex. So this is our baseline. This is before any kind of stimulation. So we wanted to see how they interact with each other. So as you can see, we have, first of all, the method I used was GPDC, which is generalized partial directed coherence. It looks at the synchronization of these signals in the frequency band. It helps us to separate these frequency bands. And then, as you can see, we have a very strong coupling between CCO and HBO in endogenic and neurogenic bands. But we don't see many significant or strong connectivities or couplings between these three nodes in the myogenic band. So in other studies, we have shown that myogenic band is mainly responsible for the global connectivity in the hemodynamics. So because it's corresponding to the smooth muscle cells, the oscillations in this layer of the vessel wall is very global. It's not affected by the regional demand for oxygen or demand for glucose or anything. So what we're mainly focusing on is endogenic and neurogenic band. As you can see here, we have some differences, for example, from EEG to HBO or from HBO to EEG. The directions show which one is leading the other one. So for example, here or here on both prefrontal cortices, we have EEG leading HBO, stronger than HBO leading EEG, which means that the demand for oxygen in the electrophysiological activity is leading the HBO, which is shown previously, actually, because the demand always leads the supply. So the other way to show these values are just the bar plots. This is exactly the same values. I just wanted to show them separately. So in the neurovascular coupling, we showed that the EEG to HBO is more than HBO to EEG, which is showing that we have the leading effect of the EEG to HBO, or the demand to supply. Especially in endogenic and myogenic band, we also have another way of showing the this connectivity or directed connectivity from the demand to supply, which is from CCO to HBO. Again, if you look at the green bars, you see that the CCO is leading HPO. So the oscillations in the CCO concentration is leading the oscillations in the HPO, especially in the endogenic and myogenic. So these are all in rest. But what about the effects of the tPBM? As you can see in this figure, I didn't report the results from myogenic band because they were not significant. But here, I only reported the significant changes after tPBM. And we can see an increase in the leading effect of CCO on EEG and leading effect of HPO on EEG, which is contradictory to what we had in the rest. So in the rest, we had EEG leading HPO, which means the demand was leading the supply. But here, we have stimulation in CCO and HPO, and both of them lead EEG simultaneously. So the supply is pushing the demand. So in some places at this, we can say that the ipsilateral prefrontal cortex, we see that again, with the effect is very local. We don't see it on the contralateral prefrontal cortex. The other thing that is important is that we see a decoupling between CCO and HPO. So we know that the tPBM, especially the lower wavelengths, have two effects. One is increase in the ATP synthesis, and the other one is the releasing of the nitric oxide, and which leads to vasodilation. So we can see here, even in the mathematical ways, that we see this decoupling between CCO oscillation and HPO oscillation, but both of them are simultaneously pushing electrophysiological activity. We see this coupling, this actually decoupling between CCO and HPO on the contralateral side, but it's weaker. And we also have it in the neurogenic band. But the other thing is that important that we see because neurogenic band is the interaction between EEG and HPO, how the neurons are interacting with the vessel wall. We see an increased interaction or coupling between oxygenated hemoglobin and EEG on the both ways. So as a summary, we can say that prefrontal tPBM modulates the information path between lateral sides of the prefrontal cortex in all three physiological subsystems. It also induces alterations in interactions between neural activity, hemodynamics, and metabolism. Also, tPBM influences hemodynamic and metabolic infraslow oscillations simultaneously, which causes an induced alteration in electrophysiological activity over prefrontal cortex. And it can be investigated as a potential intervention for reversing the aging or disease-related vasomotion malfunction. These are the groups I worked with from 2019 to 22 in Dr. Liu's lab in biophotonics and neuroimaging lab in the University of Texas at Arlington. I would be glad to take your questions. Thank you.
Thanks so much, Sadra. This is quite a